Hey, everybody, I'm going to go ahead and start. My name is Jeff Washaleski. I'm a commercial tropical fruit extension agent, and we're going to have a good workshop for you today. You see what we're looking at there. So a little housekeeping, please stay muted. You guys are doing a great job of that. We'll have question and answer at the end of the webinar. And also I'm going to put a link in the chat to a post-workshop survey. If you could fill that out, that would be awesome. So I'm going to talk to you about limes in South Florida. And the Tahiti lime is the one we're talking about. It's preferred due to its large size and juice content. It was first recognized in the US in 1875. In California, by 1883, the Tahiti lime was growing in Florida. By the 1930s, the Tahiti lime was mainly being grown commercially in South Florida, and it was grown there until the early 2000s. Today, the major growing areas are Mexico, Brazil, Israel, and Australia. So what happened? Why don't we grow it anymore? Well, in 1992, Hurricane Andrew decreased the amount of bearing lime trees from 1 million to 250,000. Also, we had NAFTA in 1994. And between 2002 and 2006, there was an aggressive citrus canker eradication program, which effectively ended the South Florida lime industry. So here what we're looking at is lime consumption in the US, and you have this row. The first row is the season, the second row is US, and the third row is imports, the fourth row is total supply. So you see in 1980, the US was leading the import, or the amount of limes that came in. By 1993, this is Hurricane Andrew. It dropped from 64 million pounds in 92 down to 13. And you see the imports is very high. And then this box over here, this is just the citrus canker, where we basically went down to zero, and that stayed until today. We have some limes now, but it's not enough to show up on the chart. This is something I sold from Freddie, so you'll see this later. This is just showing you that lime consumption has steadily gone up. Sorry about that, Freddie. So new crop choices are needed. Lychees are not bearing well due to climate, and the lychee Aranos mite is now a major concern. Avocados are suffering from laurel wilt and foreign competition. Pitaya have a host of new pests and diseases. The jackfruit market is flooded, and the USDA has been petitioned to let MMA, Sapodilla, Spondius, and Soursop. So as we'll see today, limes have some tolerance to citrus greening, but we're going to figure that out real well today in this workshop. New plantings have already occurred. These are pictures of some new plantings down at the bottom. And the new rootstocks that we'll talk about today may help to battle citrus greening and improve yield. So new crop choices are needed. There's a strong demand for limes in the US. New rootstocks should hopefully give us new hope. And limes are somewhat tolerant to citrus greening.
Okay, still, right? Yeah, okay. So I just wanted to give a little background on this lime project that we've been doing for the past 4 or 5 years, okay? I want to acknowledge the U.S. Department of Agriculture, Agricultural Marketing Service, and in particular, the Florida Department of Agriculture and Consumer Services for the grant that funded this work. And so I want to thank them for that funding stream. Okay, sorry. Okay, this is just showing you a little bit in a different way the Tahiti lime production in South Florida, the historical Tahiti lime production. And you can see we've been growing limes for, we actually before 1965. This is just showing from 65 to 2002, basically. And the green line is the acreage. And you can see at one time we had up over 7,000 acres. And then the orange line was the production in 1,000 bushels. And along the bottom, I have some code letters, F, H, C, H, and T. which F is floods, H is hurricanes, C's are freezing events. And you can see how the acreage declined dramatically in 92. This was Hurricane Andrew. And so it's just to show you that the industry has had a pretty wild history at one time. Okay. So citrus greening has been a major constraint for all Florida citrus since 2005. As Jeff went through, Florida at one time produced the majority of limes consumed in the US. Lime is susceptible to greening. There is some anecdotal information that limes seem to have some tolerance to HLB. This was just very weak evidence, but it's out there that the acid fruits do have some tolerance. And in 2014, an economic analysis did seem to suggest that you might be able to grow Tahiti limes profitably. However, we're going to discuss this results of our project and hopefully answer, or at least partially answer this question with the current status, with the current technology, and the way we're growing them, whether we can be successful. And so this is not definitive, but it is what we've done over the past 4 years. So these, just to remind people, these trees are only about 4.5 years old at this time. There are a number of putative HLB tolerant rootstocks, and they are being used. Some of the rootstocks that we're testing actually are being used as rootstocks for other citrus species in Florida. One of the other constraints that we have to be concerned with is our soil. Our soil has a very high pH, very calcareous, and so any rootstock that we use must be able to tolerate this very high pH soil successfully. And in the past, many rootstock studies have done away with a lot of rootstocks that did not tolerate our soils. So that was another thing we were interested in. There was some previous rootstock work done with Tahiti lime and on several trifoliate hybrids and some sour orange with reported tolerance, but these trials were cut short due to the citrus canker eradication program. So within the last 3 years, and I think this has changed now, there was about 60 acres of limes. And some of these plants were grafted to citrus jambury, which is also called rough lemon. This is not a reported rootstock to be tolerant of HLB, but also some people have also planted some murcots or air layered limes as well. So to address this need for citrus greening resistance or tolerance and for looking at an alternative commercial fruit crop for South Florida, we decided to put in a project to look at a demonstration planting to see if we could use some of these rootstocks and get improved production of Tahiti lime in South Florida. So we're with the plant breeders. They suggested we try 3 new seedless triploid lime hybrid selections in addition to Tahiti lime. And we have 5 rootstocks, including the most recent traditional rootstock for limes in South Florida, which was Citrus macrophylla, sometimes called Alamo. Okay, and I think that is my, yeah, this is just showing you a commercial lime planting in 2015. I believe this is one of Mark's plantings. The materials and methods, the trees were planted in March of 2017 in a 1.65 acre lime and lime hybrid scion rootstock trial. The infrastructure included microjets or micro sprinklers and high volume irrigation systems. The trees were planted at 15 by 20 for about 145 trees per acre that comes to. And they were arranged in an incomplete randomized block design with 4 blocks. Over here on the right is the identification of the scions. So we have Tahiti lime or Persian lime, and then these selections, DPI 435, 0061, and so on. We shortened their names to C11, C14, and C33. And these are the seedless triploid lime hybrids. The rootstocks included the traditional rootstock. CM is the code word for the Alamo or Citrus macrophylla, and then one called SO507, US 802, US 897, US 942. This is the layout of the planting. Excuse me, the layout of the planting. There were 239 trees to begin with. The red coloration there are some trees that were lost or died. And then the blue were some that were damaged by Hurricane Irma in 2017. So we lost a few trees. It didn't seem to matter what scion, there was no correlation with which scion or rootstock in particular. And we took, of course, we didn't take data on the dead trees, but the blue ones we took out of the data set because they were damaged by the hurricane. Materials and methods, we had an intensive nutrition and irrigation management program. The data collected including plant growth, development, flowering, and harvest, insect pest monitoring program, and comprehensive Asian citrus psyllid control program, which used systemic soil drenches and foliar applications. There was a tri-monthly HLB screening program for the disease. And then Freddie Balin did an economic data collection for the eventual enterprise budget development. And I believe that I am finished, so I will stop sharing and hand it back over to Jeff.
Okay, thank you, Jonathan. And our next speaker is Dr. Daniel Carrillo.
Okay, good afternoon, everybody. Can you see my screen?
Yes.
Oops, sorry. Okay, are you seeing the presenter mode or the real presentation?
It's good.
Okay. Okay, so our part of this project was to monitor for the Asian citrus psyllid and determine if some of these scion rootstock combinations were more attractive to it than others. But we also monitor for other pests of Lyme. And of course, this ACP, the Asian citrus psyllids or Diaphorina citri, is one of the most well-studied insects by now. It's arguably the most damaging pest of Florida's agriculture in the past years. The insect itself causes little physical damage to the tree. The problem, of course, is this, the vector of a bacteria that causes the disease called citrus greening, Juan Lombing, or HLB. The psyllids are often found in aggregations, feeding and mating on developing new leaves of all citrus species and other plants in the Rutaceae. Not all the plants in the Rutaceae, but some close relatives of citrus. Females lay eggs on the new flush, and they prefer this feather stage flush. It's very important with the dynamics, the population dynamics of the insect. Is one of the factors that is more important in the population dynamics. Once the leaves expand, they are no longer attractive to the insect, but the adults may remain feeding on mature leaves if no tender flush is available. So adults and nymphs acquire and transmit the pathogen. They have piercing sucking mouthparts that allows this insect to feed on the phloem of the host plant, because the bacteria is a phloem limited bacteria. So they feed on the phloem. And this insect has to feed an infected plant for some time to acquire the pathogen. A short probing is not sufficient for uptake. And that's important. You will see later in the management. And once the bacterium is acquired, the Asian citrus psyllid retains the pathogen and transmits the bacteria throughout its life. So it's a persistent pathogen. Here some images that show the eggs and the nymphs and these wax secretions that they produce that is very easy to observe in the field. with the nymphs growing on the growing tips of the plant, the preferred infestation site. For the management, we use the recommendations on the Free Citrus Management Guide in terms of chemical control for non-bearing trees. We relied on soil applied systemic insecticides applied as a drench to try allow the trees to get established. And as the trees grow, you have to give up those drenches and rely more on foliar sprays with various insecticides. And another management tactic that we adopted was the application of Surround. That is a kaolin particle film that is used during the dry periods, and this is a deterrent for the psyllids. Just very briefly, just to show you, this was our initial pesticide application schedule, or insecticide application schedule. As you will see, we have here drench applications alternated with foliar sprays with various insecticides with different mode of action to try to prevent the development of resistance. But it was a very rigorous insecticide schedule, very stringent. And as the trees grew, we changed this a little bit. We adopted this Surround, the kaolin film application at the beginning of the year during the dry period, reduced the number of drenches. This is for the last year. And moving forward, we cannot continue with the drenches. We're going to have to rely only on the foliar applications. So very, very strict pesticide regime. But our main objective was to determine differences in Asian citrus psyllids infestation patterns among the cyan rootstock combinations. So for this, we monitor the trees. Every time we monitor the trees, we looked at 157 trees. That is more or less 70% of the population. And we looked for presence of Asian citrus psyllids and other pests. Every time we looked at 8 trees of each rootstock cyan combination, except for this C33 US 942 that we only have 6 because we lost some trees, as Jonathan mentioned. During the first year, we monitored the trees weekly to detect when the trees first became infested, then bi-weekly, and then monthly as the trees grew and became more complicated to do the sampling. So the Asian citrus psyllid was first detected 6 weeks after planting in May 2017. And during the first 2 years, we detected the psyllid infesting 60% of the trees, at least once in 60% of the trees. And interestingly, during these first 2 years, a larger percent of the population was composed of eggs. And this is important because it seemed like in the first 2 years, we had recurrent introductions of the psyllid, but we were able to knock down the population, and we didn't have breeding populations on the trees. Seemed like the control was working. Persistent pressure of the psyllid, but we were finding fewer nymphs and fewer adults. That changed by July 2020. 98% of the trees had been infested with the ACP at least once, despite that rigorous chemical schedule, chemical application of chemicals. And the structure of the population changed. As you see here, the most abundant stage were the nymphs. And then we started to see trees that were persistently infested, that from one monitoring event to the next one, they were still infested. So established populations on the trees, that this is when the disease transmission starts to be more notorious. And by July 2021, all the trees had been infested. So clearly, even a very strict pesticide application does not protect the trees. Now, the most important part of the project was to compare the attractiveness of the different scion rootstock combinations. And throughout the 4 years, we found that the two combinations, the ones that have here the green arrows, C14 US 897 and C33 SO 507, had the least number of finds throughout the 4 years. And the highest numbers were always on this Tahiti lime US 802 and the Tahiti lime CM. Those were consistently the most attractive to the Asian citrus psyllid. But interestingly, if you see here, all 5 combinations that had the Tahiti lime as a scion were the most attractive throughout the 4 years. So it does seem like the scion rootstock combination has an effect on the attractiveness of the trees to the Asian citrus psyllid. We also monitor other pests. Interestingly, here, citrus leaf miners were even more abundant than the psyllids. So this is very problematic, especially at the beginning when the trees are recently planted. They're very susceptible to the citrus leaf miners. The older trees can tolerate the damage a little bit more, but it's very critical for the younger trees. They are very susceptible. We also found aphid thrips, weevils, moths, leaf hoppers in lower numbers than ACP and the citrus leaf miners. We also monitor for beneficial insects. Most abundant, or arthropods, I would say. The most abundant were spiders, and it's debatable whether the spiders are beneficial. They are indeed very good predators, and they feed on Asian citrus psyllid and other pests, but the populations tend to grow a lot, and they cover the canopy with webbing. So at least a couple of times we had to go around the grove removing the webbing because it was already affecting the growth of the trees. But we found also bees, butterflies, a number of parasitoids, lady beetles, dragonflies, assassin bugs. So a number of beneficial insects also. Now, to summarize, so over the 4 years, all the trees were infested at least once with ACP. So chemicals are not enough to protect the trees. We knew that before starting the experiment. We know that some combinations, some rootstock scion combinations are more attractive to ACP than others. Again, the combinations with the least numbers were the C14 US 897 and C33 SO 507. And the combination with the most finds was the Tahiti lime US 102 and Tahiti lime CM. But all 5 combinations with the Tahiti lime scion had the highest numbers of ACP throughout the 4 years. So those are more or less the results. Moving forward, what are the options to control this pest? With oranges and with other citrus, they are looking at citrus on their protective screen. So maybe that's what's going to take to be able to grow limes in South Florida. But that's another question that might not to be answered later. So that's a summary of the data that we collected. Here's my email address in case you have any question. And thanks for your attention.
Thank you so much, Daniel. Our next speaker is Dr. Romina Gaziz. She's going to talk to you about the variety performance towards citrus greening and citrus canker.
Can you hear me now?
Yes.
Can you see the screen?
Yes.
Yes, okay. So thank you for the introduction. My name is Romina Gaziz, and I'm the plant pathologist of the group. Today, as Jeff mentioned, I'm going to be discussing how well these different combinations of scions and rootstocks did under citrus greening and citrus canker pressure. So as you probably already know, both of these diseases are caused by bacterial pathogens. Both are widespread in our regions and pretty very common to see in our area. And limes are susceptible to both diseases. Now, citrus canker can be managed by using pesticides and cultural practices. However, we still don't have a cost-efficient method or management tool to really overcome the pressure of citrus greening. Now, both diseases affect the plant's vigor and productivity, and ultimately they will affect the quality of the fruit and the production of fruit. And more importantly, if you decide to grow citrus in South Florida, you will have to deal with both diseases. Now, it was already mentioned that this project was funded by FDAC. So again, thank you for funding this study. The planting was established in 2017, March. And this table shows the way the combinations were set up in a randomized block design. And we had 20 different combinations, which were comprised of 4 scions and 5 different rootstocks. We do have an uneven number of replicates, and that was just based on the availability of the rootstocks and scions combinations. At the end, we had 239 trees. As Jonathan mentioned, 5 of them died from other reasons that are not related to pests and diseases. So we ended up with 234 trees. And these evaluations and assessments are based on natural disease pressure. So the first question that we had is, we wanted to understand how the disease spreads through a new planting. And for that, we took samples every 4 months. So in the months of March, June, September, and December, we collected leaf samples and we look for the pathogen using a conventional PCR protocol. Now, I'm going to show you a couple more slides that will look like this. And what you're going to see is this table start getting green. So the cells are going to start turning green. And when that happens, it means that the tree located in that particular cell tested positive for citrus greening. So as you can see, as the time passes, the trees starting to become positive for the disease using this conventional PCR protocol. And you will also see, based on the timeline, that after a year of planting, we started getting the first positive results, the first positive trees. And by September of 2020, all the trees were positive for the disease. Now, with this, we can say that the disease spreads in a random pattern. The entire plot got infected within 3 years of planting. And so that means that all scion rootstock combinations are susceptible to citrus greening and develop symptoms. However, we did observe differences in disease severity across the different combinations. So having that in mind, we wanted to understand and assess disease severity in these different combinations, and we did this sampling in 3 years, but I'm gonna only show you the results of the last year. Something to point out is that the trees with the new flashing and that varies with the combination, can escape the foliar symptoms at least. So you can see in this picture how the older leaves show very distinctive symptoms of citrus greening, and then you see the new flash with the green tissue. Now, we evaluated the 234 trees for 4 categories. The first one was for foliage symptoms of greening. And you can see here, I'm sure you have seen it multiple times, very characteristic patches of these chlorosis and mottling on the foliage. We had a 4 number rating scale, in which 0 means that there were no symptoms observed on the plant canopy, on the foliage. And when trees were scored with a 3, means that they had severe leaf greening, meaning that more than 50% of the foliage on the canopy was symptomatic for citrus greening. Now, the second category was branch dieback, and we also had a 4 number rating scale, the same idea, 0, no symptoms, 3, severe symptoms. We also look at citrus canker because it's also a very prevalent and important disease affecting lime. And we had also these 4 rating scales, 0, no symptoms, 3, severe symptoms. Now, the 4th and last category was overall health of the tree. And this was mainly based on canopy density. And we had also a 4 number rating scale, but in this case goes from 1 to 4. 1 we gave to trees that look great, very full, very dense canopy. And 4 when you see trees with high defoliation and almost dead. These are the numerical results. We have 4 categories, as I already explained. And in the next slides, I'm going to show you this data in way of graphics. So it's better to communicate. So you're going to see a few of these bar graphs, and you see on the y-axis, you will see the percentage of trees that were categorized in the 4 different categories on the scale. And on the x-axis, you will see these different combinations. Remember, we had 20 different combinations of rootstocks and scions. Now, you will also see that these combinations are grouped by scion. So for instance, all of them in this box are from scion C11, and all of these in this box are from scion Tahiti lime. Now, based on the symptoms of citrus greening on the foliage, the scion C11 and C14 performed the best. That means that they had less symptoms, less percentage of the foliage was showing symptoms. And you will see that they have less red bars on those combinations. On the other hand, Tahiti lime and scion C33 had a lot of greening on the foliage, which is shown here as red bars on those boxes. Now, in terms of branch dieback, we found that the scion C33 followed by 314 performed the best. So you will see that these C33 and C14 have more of the blue bars and of the green bars, which are 0 and 1. So that means no symptoms or mild symptoms. And on the other hand, you will see that the Tahiti lime scions perform not very good. You can see that there's several trees rated at 3, which are severe branch dieback symptoms. However, a great percentage of these trees were categorized in rating 0 and 1. So even though there were few trees that show extreme branch dieback symptoms, there was a lot of them that were with no symptoms or mild symptoms. Now, the rootstock US 802 was the one that performed the best across all the scions. Now, citrus canker. You see that there's no blue bars in any of the combinations. That means that all the trees show citrus canker symptoms on the foliage. Now, C11 and C33, which you have here, the first box, you see a lot of red, the same as with C33. You see a lot of red and a lot of number 2s. So those were the ones that performed the worst in terms of citrus canker. And Tahiti lime performed the best in terms of citrus canker. So that means that most of the trees did not show severe symptoms of the disease. Now, there was no correlation of the rootstocks in terms of citrus canker. All of them perform more or less similar. Now, this is a very important category because this is the overall plant health. And remember, I mentioned that this is mainly based on canopy density. So in this case, C33 and C14 perform the best. You see they have no red bars inside their boxes. And again, even though Tahiti lime did have some trees that were looking really bad, in bad shape for all the rootstocks actually, since we had a very high number of Tahiti lime, it's important to say that a great percentage were classified in number 1. So more than 50% of the trees out of 78 trees that we had with Tahiti lime scion were classified in 0 or in 1, or 1 and 2, sorry. And the rootstock CM, which is the traditional rootstock, and SO507 performed the best. So in summary, even though Tahiti lime was not the best performing scion, almost 50% of the trees were scored as number 1 in overall health. Now, C33 had more than 60% of their trees scored a number 1 in overall health, so good looking trees. However, this scion had undesirable horticultural traits, which Dr. Crane is gonna mention next. Now, fruit rating. Since we really wanted to focus on Tahiti lime, those are the combinations that we harvest fruit for assessment. And we look at any kind of imperfection on the fruit. Either could be citrus greening associated, like discoloration of the fruit, or uneven ripeness of the fruit that you can see in this photo, or citrus canker lesions, which are pretty aggressive and pretty noticeable. And when that lesion dies, because the peel dies, then it can even crack as the fruit wants to grow. And also we look at other type of imperfections caused by scab, and very interestingly by bird pecking. We had a lot of bird pecking in our fruits. I'm not sure how to deal with that, but a lot of the fruit were called bad because of the bird pecking. And so we collected 14,540 fruits. It was a very busy couple of days. I have to thank my lab. We all went and with Jonathan's lab collected all these fruits. And we sort them based on their external quality, which is related to how marketable these fruits will be. And fruit with more than 10 to 15% of the area damaged for any reasons were classified as bad or cold. I really want to mention that we did see perfect fruits, grade 1, in trees that are affected by citrus greening. So even though a lot of the fruits do get damaged or are not in good shape because of the disease, it is possible to get perfect fruit out of trees with citrus greening. This is the results of the rating. And here you will see this graph showing on the y-axis the number of fruits, and on the x-axis you will see that the Tahiti lime scions with the different rootstock combinations. One thing that will jump up right away is that most of the fruits that we collected were classified as bad. So the trees do produce a lot of fruit. However, a lot of them are not marketable, at least in the fresh market and at least for external marketability. So this will have to be paired with the juice content and the quality that Jonathan will present. So in summary, all the combinations, more fruits were classified as bad, and the combination that produced the highest number of good fruits was Tahiti lime with the CM rootstock. But again, this will have to be paired with the economics and the quality of the flesh of these fruits. So with that, I want to thank you for listening. This is my email if you have any questions. And thank you to the funding agencies and to my lab that help us with the fruit harvesting and the fruit sorting. Thank you.
Thank you so much, Romina. And everybody remember, at the end, we'll have a chance to answer questions. And I just put a link to the post workshop survey in the chat, if you could answer that at the end. Our next speaker, Dr. Jonathan Crane, he'll be talking about the performance of Tahiti limes and others, new versus old rootstocks.
Thank you, Jeff. Okay. You can see the screen?
Yes.
Thank you. Okay, so I'm going to go over some of the results from the trial. Okay, so as I mentioned, oh, let me back up. Actually, yes. Okay. Again, this is, I killed this one time already. All right. Okay, sorry about that. So first thing, again, this is how the treatments were, the different rootstocks and the different science, okay? And this is just to show you a summary of the irrigation practices during this trial. And you can see that we used our micro irrigation system during 2017, 18, and 19. We then had some substantial problems in 2020 and 21 with that system. So we switched over to a high volume under tree system. It worked sufficiently. The downside of it is that you were getting the foliage wet. The upside is that it did work as a backup. This is to show you some of the fertilizer practices that we implemented. And this stems from a combination of old-time citrus practices or lime practices and suggested practices based on the newer work that's been done on oranges in the state. And so we did use some liquid NPK to get the trees established the first 2 years. We also used a slow release material. So the idea, the strategy for the fertilizer program was to have the roots and the leaves to have access to nutrients for as much of the time as possible, so that at any time that the roots were active or that the leaves could take up nutrients, they would have nutrients available. So the slow release materials, that was the purpose of that. The granular materials, the 839, for instance, when you look at that more closely, part of it is actually slow release and some of it is more quickly release. And that's why we used some of the 839, because that has a more quickly available material as well. And so you'll see the amount of fertilizer we used in the first column, the number of applications during the year. So we split, you can see that many of these applications were split. And then the total amount that was used of that particular material on 1 acre. I calculated it back as 1 acre of these materials. So as an example, you can see the sequestrine iron, which is a soil drench. We were using 2.3 pounds. We did this 4 times. For the year 2017, we used 9.2 pounds. You'll also note that as the trees matured from year 1, 2, 3, and 4. Especially in year 2 and 3, we increased the amount of fertilizer per tree at each application. And also in some cases increased the number of applications in a year, for instance. In 2020, we actually reduced our nitrogen inputs to some extent, and we tried to increase slightly our foliar minor element sprays and increased we used phosphite, and here you'll see phosphite. Phosphites or phosphonates don't actually, they're a phosphorus compound, don't actually apply phosphorus to the tree that's in a usable form. What it does do is help to protect the root system from root rotting fungi. And that was the strategy. As you recall, we had a hurricane in 2017. And so we started using these phosphonates in an effort to help protect that root system. And then more recently, we have been adding a micronutrient soil drench material mixed with a sulfur compound to lower the pH. And we've been doing that this year to see if this does not enhance. So you can see the strategy now is we're applying the minor elements, not just to the soil, but also to the leaves and the soil. Okay, so this says cyan comparison eliminations, and what I'm getting at is I am going to talk mostly about Tahiti lime cyan. And I'm going to give you some reasons why we're not going to go into great depth, at least at this time, on these other cyans. The C11, C14, and C33 are seedless triploid hybrids. When we did the evaluations, it included some informal tasting as well, and we found that C11 and C14, were sour. Well, so is Tahiti lime, but the taste was only fair. And then when you consumed C33, in our experience, we found it to be bitter and of a poor taste. Now, some of this may be that we did not pick these limes at their optimum stage. However, I will say, in our defense, we harvested these various lime hybrids at different stages to try to find the stage at which they were good. We did not find a good stage, at least not yet. The Tahiti lime or the seedless, it is sour, but as you guys know, at least in my view, it has an excellent flavor. So that was one thing. Secondly, had to do with the growth habit of the trees. C33, I put down there as a deal breaker, and that's because it had thorns, as you can see in those pictures, that were 1 inch to 2 inches long. That, I don't know who you would get to pick that fruit. I even bought Kevlar gloves and Kevlar sleeves to try to pick that fruit, and I still got impaled picking that fruit. The other bad side about C33 is most of the fruit is concentrated on the inside of the tree. So that means you have to go buy a whole bunch of thorns to get to the fruit. So C33, in my view, right now is, if it was worth its weight in gold, maybe, but it's not even close. Other thing that we took data on was rootstock sprouting. And this is important. Why? Because if the rootstock below where it's grafted sprouts out, this can end up taking over the cyan tree and interfering with production of the fruit that you want to produce. That's one thing. The second thing is if you don't remove it in a timely manner, and that means mostly when it's small, even smaller than this, if possible. When you remove it, it causes a wound. Well, if you let it get really large, you cause a really big wound. Then you can get rain splash, which puts spores up onto that wound, and you end up with a stem rot. And so now you have a second problem. So rootstock sprouting can be of great concern. The old time rootstock that we used, rough lemon, in the past, one of the reasons why people stopped using rough lemon was because of the rootstock sprouting, which was persistent with rough lemon. We looked at this for these various rootstocks as well. And here, what I'm showing you is we went ahead and took data from 2017 through March of 2018. And this is the percentage of rootstock sprouting over this 12-month period. And, you know, US 802 really stands out, although 897 and 942 are also standouts. Probably the two best ones, and certainly the best one, was Citrus macrophylla, and the second best would be SO507. And even now, let me, excuse me, sorry. Let me, even now, US 802 and 942, we're still seeing some rootstock sprouting. So it has persisted as a problem with them. If we look at early flowering, we took data on that at the beginning. Well, we followed it throughout the trial, but at the very beginning to see if they gave us a hint on maybe they could come into production a little bit early. Limes can, you know, in the past, before greening, you could put in air layered limes and they'd start producing fruit in year 2, and grafted trees basically in year 3. And so we looked at this to see if there was some indication, and again, it looked like C11 and TL and C14, you know, was having some early flowering, which may result in early production. If we look at the crop production comparison, so here I'm comparing all 20 scion rootstock combinations. Here's TL, here's C11, C14, C33. And this is from the third harvest, which the trees were about 3.5 years old. And what I did was I took the harvest data and recalculated it into if I had a grove with 217 trees per acre. And the reason I did that is that even though we only had 145 trees per acre, really one of the newer strategies for producing citrus in Florida is to put in high density plantings, with the idea is that you want to get the trees planted, get them up and going really, really quickly, start producing quickly, and get them into this early fruit production. And so our trees are not to the stage where they were competing for water, nutrients, or sunlight. So they have no closed canopy within the row or even between rows. So it seemed justified to go ahead and recalculate this on a more high density planting to give you a feel for what this might look like. Having said all that, you can see that by far and away the Tahiti lime, especially on these 3 rootstocks, was producing much more than the C11 and C14. And although C33 had some substantial production, compared to the others as well, again, the thorns were an issue. So I'm going to focus on the Tahiti lime for this talk. What I'm showing you here is some of the growth patterns or phenology. And what you have here are two figures here. This is the first figure, and this has to do with the shoot or leaf flushing pattern on the limes, on the 5 rootstocks, all with Tahiti lime. And the scale goes from fewer trees to more trees flushing. So, if you had 10 citrus macrophylla trees, this would mean all 10 are flushing. Here would mean none of the trees are flushing. And as you can see, the flushing pattern was generally the same among all the Tahiti lime and rootstock combinations. And you can see there was a period of time where there was very little flushing, but you also note that during the year, there was almost no time that during this 1.5 years, almost no time in which there were no trees flushing. So these trees are constantly flushing, and this also plays into the concern that the citrus psyllid is attracted to this new flush. And we knew from historical data that Tahiti limes would flush up to 10 times a year. So this is not, a revelation, but it's confirming that they flush a lot. If we look at the flowering pattern, again, it goes from fewer trees at the bottom or no trees flowering to all the trees flowering of a particular scion rootstock combination. Again, the pattern And you can assume that these patterns are being dominated partly by genetics, but also by environmental conditions and perhaps some by cultural practices. And again, you can see that there was some flowering present on all these combinations. Most of the time, there was some brief periods where there was very little flowering. Okay. If we look at the fruit production, and this is for trees, there were 4 harvests. The first harvest really started in September 2019. We also harvested, and let me just go back. You know, if you look at the lime industry, the history of the lime industry, our major production on the trees was June through September. However, we also did have production the year round. It's just that it was less or much less depending on what time of year. So the main production period is generally June, July, August, September. We did have a light crop in November of 2020, and then the more recent crop was here in July this year. Okay. So several things. One is there was no significant difference among the rootstocks within the first 3 harvests, right? Doesn't matter. There was no significant difference in how many Tahiti limes they produced. With the 4th harvest, there was some difference. And whether you're looking at lime fruit produced per tree or per 217 trees, it's the same graph. I just wanted to show you that. But you can see Citrus macrophylla and SO5 and US 897 were basically the same, but TL was significantly greater than US 802 and 942. However, if you compare this to yields for 4-year-old trees in the past, prior, and I hate to keep referring to this, but prior to HLB, to citrus greening, our yields would be somewhere between 33 and 60 some odd % below what it was historically. So if you then look at all 4 harvests together, so you combine them all, SO5 and trees on SO5 and Citrus macrophylla had significantly more fruit than those on 802. It wasn't different than 897 and 942. Fruit quality. So in this last harvest, we, as Romina talked about, we had cull fruit and good fruit, and we tried to differentiate between the two and took data on that. And so in general, all the rootstocks produced more unmarketable fruit than marketable fruit. And I'll talk a little bit more about that in a minute. But this does suggest that it's not entirely a rootstock effect, but perhaps a tree management effect. So in other words, better citrus canker management, perhaps, at least at this planting, bird control. Bird control has not been an issue historically, but definitely in our planting, I'm not sure if it's because of what we're surrounded by or not. Maybe it's because we're surrounded by a diversity of grove types and fields. I'm not sure. But as you can see, there was, significantly more culls with those on CM than 897 and 942, for instance. And here's some of the issues, and Romina showed some of the same photos. And this is probably actually, canker, and the peel died and it split. As I put down maybe mites, saw very little indication of any mite problem. Canker, peel yellowing, and just to let you know, there's been some very good work done in Mexico on Tahiti lime. And peel yellowing was associated and correlated with citrus greening and is an issue. So citrus greening is having an effect on the peel quality and the peel color and peel development, basically. The other, as Romina mentioned, had to do with the pecking of the peel by birds. So if we look at individual fruit quality, or the mean, actually, the average. And this is the data taken from 4 different harvests. There was no significant difference among the rootstocks in fruit diameter or in the fruit lime weight. What I will comment on, though, is that previously the minimum lime diameter for harvesting limes commercially in South Florida was 1 7/8 inch in diameter, which is 1.88 inches, is another way to say it. We also, as part of the lime marketing order, just before the lime industry was destroyed, had put in effect a 2-inch lime diameter minimum for limes produced during the summer. And so you'll see here, all the limes that we harvested basically were above this minimum 1 7/8 inch. And so we actually did go out and purposely, the limes that we selected for this diameter and fruit weight analysis and juice content were limes that were of at least 1 7/8 inch in diameter. The other thing too is in the fruit weight. Normally, the individual weight for fruit was previously 1.9 to about 2.2 ounces, and you can see we've exceeded that as well. But again, there was no significant difference, even though it looks like there was some differences. More importantly is the juice content, because previously we had a marketing order and limes had to have a 42% juice content by volume in order to be legally marketed. We calculated our juice content a little bit differently because it was a faster method. We used the weight to weight method, so the weight of the lime versus the weight of the juice. And when you do that, there's generally the figures come out with about a 3% higher volume of percent juice content than if you do it on the volume basis. Having said that, again, there was no significant difference among rootstocks. I already mentioned that previously the minimum juice content for harvesting limes was 42% by volume. The measurements on a weight basis are usually 3 to 10% higher, but you can see we are not 3 or 10% higher than 42%. Therefore, citrus greening may be responsible for this slightly lower juice content. And indeed, this work out of Mexico also found that citrus greening had a negative effect on the juice content of the limes. And that makes sense. When you think about this bacterial disease, it is parasitic on the tree. It is taking the resources from that tree. So the tree is struggling to produce fruit and to fill the fruit juice vesicles. So conclusions, the vegetative flushing and the flowering patterns of Tahiti lime on the 4 rootstocks tested was similar. The fruit production on all 4 rootstocks increased dramatically by year 4, and that wasn't unexpected. However, even for 4-year-old trees on the citrus macrophylla, rootstock yields were below historical yields. Over the 4 harvests, there was some significant difference in yields among the rootstocks, with CM, SO5, 07 and US 897 were similar, but greater than Tahiti lime on US 802 and 942. However, on the last harvest, the SO507 and the CM produced significantly more fruit than 802 and 8, but was similar to 897 and 942. There was no significant difference in the amount of marketable fruit produced among the rootstocks tested, although we had a lot more cull fruit. The CM had more cull fruit than 897 and 942, but the amount of cull fruit was similar for CM, SO507, and 802. We've already mentioned the fruit quality issues having to do with canker, peel yellowing, bird pecking. I said maybe mites. There was no significant difference in the fruit diameter, fruit weight, and juice content for the limes on any of these rootstocks tested. And I'm going to stop here right now and hand it back to Jeff for the next speaker.
Thank you so much, Jonathan. And Manji put into the chat that you can use tree wraps to reduce rootstock sprouts, just so everybody sees that.
And actually we did go to using tree wraps. And that's really what stopped most of the sprouting. We did use tree wraps. After that 2018, the last measurement, we put tree wraps, and that did stop most of it, not all of it. So that Manji's correct. Yeah, you can use them.
Okay, excellent. Our next speaker is Freddie Belain. He is going to talk about, is Tahiti lime industry in South Florida financially viable? So what we've all been wanting to hear.
Okay, good afternoon. Just 1 second to get my screen. Oops. Okay, so can you see my screen now?
Yes, perfect.
Okay. So the question is, Tahiti lime industry in South Florida financially viable? Let's go over that question. So this is a brief outline of my talk. First, some background and market information. Then I will go over the economic analysis of the feasibility of Tahiti lime production, and finally some concluding remarks. So let's start with some background and market information. So this graph shows Florida Tahiti lime acreage and yield from 1980 to 2001. And we can see that from 1981 to 1992, acreage was over 6,000 acres. Then in 1992, we had Hurricane Andrew. It hit the growing area, and then acreage decreased by 60%. So by 1993, area was close to 2,000 acres. Then the industry was on a recovery mode. But then in the early 2000s, we had the citrus canker mitigation program, and all the residential and commercial citrus plantings were eliminated. Also, we can see that from 1980 to 1992, average yield was about 359 bushels per acre. That was before Hurricane Andrew. Now we can see the production and imports total supply from 1980 to 2002. And it's possible to see that from 1980 to 1990, Florida was the main supplier of Tahiti lime. But then we see that in 1992, as we got hit by Hurricane Andrew, imports started to increase. And also in 1994, the NAFTA agreement went into effect. So we can see that after that point, imports of limes increased significantly. So now we can see over the last 19 years how imports of fresh Tahiti limes have grown by more than 3 times. So back in 2002, imports were about 2,024,000 metric tons. And last year, imports were about 680,000 metric tons. And Mexico is the unchallenged supplier. We can see that more than 95% of the total supply of fresh limes comes from Mexico. This graph shows the average monthly Tahiti lime imports for the last 5 years. So it shows some seasonality. And we can see that May, June, July, and August, imports are over 55,000 metric tons. So most of the Mexican crop comes at a time when the bulk of Florida crop is being harvested. So remember, Florida, the main harvest season for Florida is from June to August. So we have to compete with higher supplies from Mexico. In contrast to what happened in the fresh fresh lime market import, the market for US processed lime imports have been stagnant for most of the time under consideration. Since 2016, we have seen some growth in the import of processed lime products. And by 2020, imports reached close to 600 metric tons. That means that consumers are starting to put some attention to processed lime products. And also at the same time, it can mean some opportunity for second grade fruit in the domestic market. Next, we can see US per capita consumption of selected citrus fruits for the last 10 years. We see that consumption of oranges has trended downward from 2010 to 2017. Then it bounced slightly and reached 9.2 pounds back in 2019. Consumption of limes and lemons have trended upwards, particularly per capita consumption of fresh limes has increased by 70% from 2.5 pounds in 2010 to 4.7 pounds in 2019. And there are several reasons that help to explain that growth in demand. So first, we have an increase in the US Hispanic population. We have also seen the growing popularity of Mexican food, and also limes are used in other ethnic cuisines. Because of the low fruit volume shipped, price information for Florida limes is very limited. This is the latest report we got from the Agricultural Marketing Service, and this goes back to last week of June 2017. And the important thing to see here is that there are no price differentials for fruit with a bigger size. If we see, for example, the prices for a 40-pound cartoon for 150s and 175s, that's about the same. There is a slight decrease in price for the 200s, and this is just 50 cents below. But in general, we can tell that there is a premium for larger size fruit. Consistent with the seasonality in imports, we also have the wholesale prices also follow a seasonal pattern. And the graph shows Miami monthly wholesale average prices for the last 3 years. And we see that prices are higher in March, usually get $35 for a 40-pound cartoon. Then as we get more supply from Mexico, prices start to decrease, and we reach a low of $13 per box in July. That's the lowest of the season. And another thing is that I have shown the prices for two different sizes, 110s and 200s. And again, there is no real price difference. Even if we look at May, there is even a slight discount for bigger size fruit in the Miami market. Okay, so now we will talk about the economic feasibility of Tahiti limes production in South Florida. And let's first take a look at returns before and after the citrus eradication program. So the table on the top shows the returns before the citrus canker eradication program. And we have considered the first 4 years of the crop. And we see that we have positive returns in year 3, and by year 4, after we consider expenses, income per tree is about $4.48. Now, in the table at the bottom, we see that the GL situation under the current GL under both the HLB and the citrus canker disease. And we have that marketable GL is about half of what it used to be. And we don't see in the first 4 years, we don't see any positive return for the trees. Now we face higher production costs and of course, lower yields. So this is for 4 years using our experimental data. So this shows clearly the situation before and now. So now the question is, what would happen over a 20-year period? So this data, this analysis is specific for the TL-CM Zion rootstock combination. And on the first row, we have average yield before the tree eradication program. And now below in blue, we have the current average yield. So we can see again, current yields are about half of what it used to be. Now we have to follow a more intensive fertilization and pest control program to control the diseases. And price now is $25 per bushel. Harvest cost is about $6.50 per bushel. And the analysis was done using a discounted cash flow analysis over a 20-year horizon. The discount rate used was 5%, it's a baseline. The UST note yield over a 20-year period is 1.85%. So if you were to start up a new Tahiti lime grove, at the end of the 20 years, you will get losses. So it's not feasible. So your losses will be about $23,000 per acre. If we look at the benefit cost ratio, it means that cost outweigh the benefits. So the project is not feasible, it's not possible. So this is just assuming average yield. But now, what if we include, if we make the analysis more realistic by incorporating production and adverse weather risk? So in the first row, we have low yields, and in the second row, high yields. For example, for a 4 years old tree, based on in our experimental data, the tree can have a yield of 0.57 bushels and could be as high as 1.5 bushels. And then to account for a severe weather event, we consider a 10% chance that a hurricane will hit South Florida, and that might result in a 50% drop in yield. We use the same production cost and prices as before, and also conducted the same discounted cash flow analysis. And then under this more realistic scenario, where we have varying yields and the possibility of getting a hurricane, the losses could be could be like $40,000 per acre. And again, the benefits cost ratio shows that costs are higher. So costs outweigh any benefits that we might get from planting, from starting a new planting. So what does this analysis tell us? So demand for Tahiti limes keeps growing, that's right, especially during the summer harvest season. But at the same time, that's when most of the crop from Mexico lands the US market. There is market opportunity for high quality fruit that could be of interest for the hospitality industry, and also there could be a market opportunity for local fruit that could appeal to consumers, Florida consumers in specialty markets. Also, we found that at the wholesale level, we don't get premiums for larger fruit. We might compete on the basis of quality, but at this time, the market doesn't pay any premiums for larger fruit. Yields are low, about 50% less of what it used to be because of the disease pressure. Remember, we are dealing with 2 diseases at the same time. And with current prices, cannot make up for low yields with current planting material and crop management practices. This is not to say that everything is lost, it's just that we need to improve from the technology side to make it profitable. And that's all that I have for today. You have my contact information if you have questions.